Okay, as I hopefully have been emphasizing enough here, CRISPR-Cas9 is really the way to go as far as genome engineering is concerned. At least that's what most people think, including lots of companies that are trying to sell you their products as far as doing genome editing is concerned. Another quick thing is some of you may have seen in the lecture notes, I've set aside some time today to do course evaluations. And that means that since I shouldn't be here while you're doing those, office hours will be held over in SRTC in the conference room up on the 4th floor, starting when class is over, which will probably be about 11:05, 11:10 today. If you've already done your course evaluations, you can come over and grab me then immediately. Yeah.
Is that the same as what was the online link?
It should be the online link that you got in your email, with any luck. I don't get my own evaluations. Gee, I could just evaluate all of you, but that's what the final exam's for, right? Okay, any questions about that at this point? No? The exam is on Wednesday of finals week at 10. So, and we can set up extra office hours if people would like then in that week as well.
Do you see those evaluations?
Pardon? I will see your evaluations after I have turned in all the grades, and they are anonymous, so I have no idea who you are, unless you put your name in them, which I think actually gets striked out anyway. So, but it's after all the grades have been turned in that I actually see those. But they are extremely useful to me in terms of how I'll be teaching this course again, hopefully how I'll be teaching courses in future. So please let me know what you think about this. And one thing in particular I'd like you to tell me about are our clicker questions. This one's way too wordy, I know, I'm sorry, but I wanted to make it as obvious as possible. With any luck, if an mRNA with 2 different AUG sequences is regulated by leaky scanning, what are the maximum possible number of non-identical proteins produced? Only 1 protein can be produced by the messenger RNA, 2 proteins can be produced, but most must have the same C-terminal sequences. 2 proteins can be produced, but must have the same N-terminal sequences. 4 proteins can be produced, but at least 2 must have the same N-terminal sequences. 4 can be produced from the mRNA, but at least 2 must have the same C-terminal sequences. Now it's already 30 seconds worth of the question. Quick show of hands if we want more time. So I'll start again once it's done. Okay, I'm going to start again up here. So please re-vote. Yes, you do have to vote again. Vote early, vote often. Only one counts. 10. Okay, so the vast majority of you think two proteins must have the same C-terminal sequences. That is a good start, but unfortunately, only one person got the right answer, which means it's probably a bad question. At least it's better than D, which was completely wrong and nobody guessed. Leaky, what does leaky scanning mean? What does leaky scanning mean? You can use different AUGs, right? So if the AUG is in the same reading frame as the downstream orth, the main open reading frame, what does that mean? Two proteins, same C-termini. If the first AUG is in a different open reading frame, what does that mean? Two completely different proteins that you could get through that process, because your reading frame is shifted. AUG, depending, that's nucleotides 1, 2, 3, and I guess I didn't do a good job of explaining reading frames, which is a very important point, which is why I'm covering it again now. Reading frame means you take nucleotides 1, 2, 3, the next codon is 4, 5, 6. If your AUG is moved one nucleotide, now instead of 4, 5, 6, you're 6, 7, 8. Which could be a completely different amino acid. If you're at 7, 8, 9, that could be a completely different amino acid. So we'll talk a little bit more about this, actually probably on Wednesday. But any given double-stranded DNA sequence, you can have 6 different peptides coded from that. Each of the 3 open reading frames on one strand, each of the 3 open reading frames on the other strand. So it makes 4 because you've got 2 possibilities, which most people seem to have gotten, with the extension at the N-terminus, but that's only if it's in the same open reading frame. If it's in a different open reading frame, now you have 2 other ones. So that gives you 4, 2 of which have to have the same C-terminus. That makes sense? No? No, not at all? So AUG, 2 different AUGs in the same open reading frame. That's what most people seem to have gotten, or they were just listening to people who said loudly that it was going to be 2. So that's, regular open reading frame at the end, right? Starts in AUG, methionine, and goes. If you have another AUG which is upstream, as long as it's multiples of 3 nucleotides upstream, you'll have an N-terminal extension on that next protein. But the C-terminus is going to be identical because you're in the same open reading frame. If, however, that first AUG is in a different open reading frame, the rest of the protein is going to be completely different downstream of that. So it all depends on where you start, what open reading frame you're using, and what peptide you get. Yes?
I understand that you could have 2 sets and 2 sets, but you can't have both 2 sets at the same time.
No, what is the maximum possible number of proteins that can be produced? That's what the question says. What are the maximum possible number of non-identical proteins? I thought that was pretty clear. Obviously, it wasn't. Again, really obviously it wasn't. So, which means that it gets normalized away anyway, so it doesn't really count, except for that one person who gets an extra point, and I don't know who it is. So which of the following gives you the most fold purification of your favorite protein? Oops, no one's actually voted yet, so that won't help. Differential centrifugation, ion exchange chromatography, size exclusion chromatography, hydrophobic interaction chromatography, or affinity chromatography. Hopefully we'll have a few more. Yes?
What do you mean by most fold purification?
So best purification, highest resolution. So starting out with a whole mixture, how do you get the purest thing out from that whole mixture with your chromatography? But there was enough space in the slide to put that whole thing up there.
Accidentally forgot my clicker. What should I do? So just for people.
Yeah.
Okay.
10, 5, liftoff. That looks a lot better. I'm happy with that. Are we happy with that? Yes, good. Because the answer is E again. Stedman's got to stop having E for all the answers to his questions. It won't be that way in the final, I promise you. Shucks. Okay, so those of you have to leave, good luck. The rest of you, we'll continue to talk about the protein purification and analysis methods today. And then probably before we get to much in the way of DNA methods, I will run away and you get to tell me what you think of clicker questions that only one person in the class can answer properly. So again, we'll continue to talk about proteins. And in fact, the first couple of slides are actually about those particular examples of chromatography. Talk a little bit about gel electrophoresis. This is an analysis method, much more so than a purification method. And then what people do once they've found these kinds of things in terms of how do you determine the structure of a protein, which will hopefully determine the function, but unfortunately from structures you can't always determine what the function is going to be. Bioinformatics or computational biology, this is actually very much the first step that people usually take these days because doing nucleotide sequencing is very easy, very fast, very cheap. Doing all of these protein methods is very slow, very annoying, and very expensive. And you want to work questions about the annoying part, ask people in my lab because we do a lot of this stuff. And then we'll talk a little bit about some protein-protein interaction assays and how people look at how these things interact. And so how people come up with those cartoons like we've seen in terms of transcriptional regulation. You've got all the mediator and all the general transcription factors and all these things associated with each other. All your transcriptional activators associated with each other. How do people know? How do you tell that? Those are some of the protein-protein interaction assays that we'll be looking at. And then with DNA, we'll talk a little bit about how you do, and this should be DNA gel electrophoresis actually over here. Restriction endonucleases we'll probably end up getting to on Friday. And basically the notes that I posted last night for today will be basically the same ones that we end up using on Friday as well. So if you printed them out, just bring that second set with you. So we'll start out talking a little bit more about chromatography. This again are your columns that have a matrix in them that will change how your favorite protein comes off of them. And your favorite protein here is basically what's listed down here at the bottom in blue. This would be, for instance, your DNA binding protein. How do you know that this is the protein you're interested in? Because you figured out some kind of activity. So that activity could be a DNA binding activity through a electrophoretic mobility shift assay. It could be some kind of enzymatic activity. It could be some protein-protein interaction activity. Whatever that activity is, that's what you're using to try and decide which of all the proteins here, which are just more or less shown by this red line up the top here. This red line represents protein concentration all the way coming off of your column. And so one of the things that should be really obvious here is that there's still kind of a nasty mixture. If you have one nice peak, that will be nice, but you've got this whole mixture of different things here. And this is a classic example, not actually a real one, but pretty similar to what you often see. Ion exchange chromatography, you have some proteins that aren't going to bind at all to the matrix, and these will be those that have the same charge as the matrix that you're using for your ion chromatography. And then with your increasing salt concentration, you will have those that come off the column that have very little charge, and then those that come off at a very high charge at this end. So this process, the flow through, that's what we have here. It's when you have something which doesn't bind at all to your column, and then a gradient of increasing salt concentration, which will get, again, the proteins that have a small amount of charge here at this end that have a large amount of charge at this end. What you do is off this column, usually you don't have a nice pretty color that you can see, so you'll just collect, this is milliliter 1 to 2, this is milliliter 2 to 3, etc., all the way across here, and test every single one of these and say, oh, look, milliliters 18, 19, and 20 have the activity that I'm interested in. So usually what you'll do is you'll take these, which have this set of proteins in them, put them onto your next column. And so this is your example of gel filtration or size exclusion chromatography. Here you'll notice there's no gradient, but that's just because you're putting your mixture of proteins at the top and letting them run through. And those that are the biggest come through at the beginning. Those are the smallest come through at the end. And again, you're looking for your activity. A couple things to look at here. One is you'll notice that instead of it being in 2 fractions here, now you're in 4 because it's a dilution process, also known as a sizing column. If you look in the literature, here's somebody talk about it, a sizing column. Again, it's just separated on size. Yes, your question.
What's the gel compound?
Oh, so the gel compound here and the gel filtration chromatography, those are the beads, the matrix beads that we talked about last time that have little holes in them. And very often they're made up of agarose or some kind of gel. Okay, so we've done ion exchange, we've done gel filtration. What's the best way to do these things, as we had in our clicker question? Affinity chromatography. So here, and what these lines mean is that this peak in your flow through is usually absolutely huge, because the idea here is the vast majority of the proteins that you put onto this column don't interact with your matrix at all. So they're all going to be in this flow through fraction. And then your elution should be something very specific. So we'll go back to that DNA binding protein. Here you have a specific DNA bound to your matrix that your protein is going to bind to. And then you put in either very high salt concentrations, or in the easiest case, you just put in a whole bunch of that DNA, which is no longer bound to a bead. Now the protein that was bound to the beads will bind to that other DNA and come off the column. And one of the things that you really like to see in a case like this will be that your activity corresponds exactly to the protein which is there. This protein, by the way, the red line, usually corresponds to an absorbance. Usually people use 280 nanometers. That's where you have some of the amino acids, particularly the aromatic amino acids, which are going to absorb. So it's a really easy thing to follow, but again, it's just protein. It doesn't tell you anything about activity. So you need to do these activities again. And this is just an example over here on the side. This would be the kind of mixture of proteins that you put onto one of these columns in the first place. These are the fractions that come off of, from this mixture, these proteins come off of that affinity column. This band is basically almost invisible over here. So you see it's a very, very small amount, which is now much more concentrated here. And this is that fold purification. Same thing is true here, same thing is true here. And then you take these mixture of proteins, bind them to another affinity column, and comes off here. Anybody know what these proteins are that are binding to these cyclins? They're cyclin-dependent kinases, not surprisingly. And so they're binding with the affinity of those protein-protein interactions. So if you're looking for something that binds to a cyclin, one way to do it is to make a column, put up on that column something you want it to interact with. So this is great if you happen to have some very specific thing that you know your favorite protein is going to be binding to. Most of the time you have no idea what it's going to be binding to. It's partly why you're purifying the protein in the first place. So what people generally do these days is they will take the protein that they're interested in and hook it up to something that allows it to be purified with one of these affinity chromatography processes. And very often this will be 6-histidines, as we mentioned last time, that will bind very tightly to metal columns. You could also have here something which will bind specifically to an antibody that you have lots and lots of. And so here you have your protein and then this extra little tag at the end, or the fusion, because you've added this tag. This tag could be at the C terminus as it's shown here, could be at the N terminus as well. This now tagged protein with an epitope tag, that's just what an antibody is going to interact with. You can now purify this because you have an antibody that you can attach to your beads and do affinity chromatography. If you happen to have this with an antibody, now you can also use an antibody that will react with that tag, and it should hopefully be only your favorite protein that has that tag on it. So the antibody to the tag will actually also be guilt by association interacting with your protein. So this is the way that most people do protein purification these days, these fusion proteins, because it makes it a lot easier. If you don't have a fusion protein, then you have all these extra steps you do, ion exchange, etc. So how do you get these pretty looking gel electrophoresis patterns? This is just taking advantage of the fact that proteins are charged, and we'll see how to get particularly good charges in just a second here. And basically all that you do is separate your proteins in an electric field with some kind of separation matrix. And in this case, it's usually a polyacrylamide gel. Sometimes you use agarose, etc. It depends on what exactly you're trying to separate. It depends on the pore sizes, how big the holes are in your gel, how small the holes are in your gel. And basically you put these under an electric field, and they will migrate from cathode to anode down here in the bottom. How do you get your proteins to move in this electric field? Well, if they happen to be positively charged, they'll go ahead and move through directly. However, as we saw from ion exchange, proteins have variable different charges, and it's all going to depend on what the charge is on the outside of your protein. So what people discovered, and particularly this was Ulrich Lemley, I don't know, back even before I was in graduate school, early 80s, was that you treat your proteins with detergents, and particularly charged detergents. What you can do is get a uniform charge to mass ratio, and also take your protein and pull it apart so that all of the hydrophobic, hydrophilic pieces of your protein are exposed. And so the idea here is that most proteins are really heterogeneous in terms of what their surface charge is. They'll have maybe a positive charge patch over here, a negative charge patch over here, etc. What you want to do in order to, if you just want to separate based on size, and some people want to separate also based on the charge, etc., what's called a native gel electrophoresis, but mostly you just care about the size. So to deal with that, you treat with a detergent like this one, sodium dodecyl sulfate, charged, but with a really long hydrophobic chain on it, and basically what that does is it'll pull your protein apart into an unfolded form. Some proteins have disulfide bonds, so you put in a little bit of this reducing agent, which will break all of those disulfide bonds, and you heat up your protein. So this is a way you can get your protein, A, to have a uniform charge, or charge density, more to the appropriate point, but much more importantly, you also solubilize it. Many proteins are insoluble, and if you have an insoluble protein, you try and put it on a gel, it just sort of sits there. And so this gives you solubility, but also a uniform charge density. So you do this, and this is the cartoon way of doing this. Here's your protein that you start out with. You treat it with sodium dodecyl sulfate and a reducing agent that'll pull everything apart and give you a uniform charge density, which means that now you're separating just based on size rather than size and charge. So this process, heating it up with a detergent and reducing agent, it's also called denaturation, pulling apart. So native is the normal structure, and denaturation is getting it away from that native state. And it gives you the approximate molecular mass when you separate these in a polyacrylamide gel. But usually, just like the case with the proteins coming off of your column, you can't visualize these proteins after they're on a gel. You have to have some kind of way of detecting them. And usually this will be some kind of dye, a stain for staining your proteins. So what does this look like? Here's a nice example of a whole purification process. Up at the top here you have cells that you've just broken open, which have a whole mess of proteins in them. Actually, this is a very clean preparation here with a number of proteins in it. You'll centrifuge this mess of proteins, and you'll end up with a different set, but still highly impure proteins. Then you'll do different chromatography steps and end up with a, as far as you can tell here, homogeneous protein. So you started out over here through various centrifugation, column chromatography, finally a pure protein here. What do you do with your pure protein? You can do structure, you can do biochemical assays, you can make antibodies, you can do all kinds of fun things with your favorite protein. But if it's in this whole mixture, you don't know which of these different proteins, and all these bands here are going to represent a different protein, actually have the activity that you're looking for. So it's very important that you get this single protein out here at the end. Yeah?
How do we make this singling out specific? How is it not random?
So how do we make this, the question is, how do we make this singling out specific? On the last step here, this is an affinity chromatography, and so you have something that this particular protein associates with. Back here, you have to know which of the fractions you want to collect. So you have to have some detection method for looking at it. In this particular case, you've got so much of this protein to start with, you can just do SDS page, your gel electrophoresis, and say, oh, I'm just looking for this band. And so I'll take the fractions that have this band in them. But you could have a DNA binding assay, for instance, and say, okay, these are all of the proteins that are binding to DNA. One of them is the one I want. Okay, I'll look here. These are a whole bunch of proteins that bind to DNA. This is the one I want, etc. Yeah?
So these are all kind of like pre-steps to be able to work with the specific protein and then play with it after all of these steps. So these are like getting ready to do more.
Yeah, so this process is, you're exactly right, basically going through a whole process in order to get finally something that you can work with. And so, yes, it's a whole involved process. But this is exactly how people found all of the transcription initiation factors, how people found RNA polymerases. It was going through this whole arduous process and then finally getting one thing that they could look at and study. Yeah?
I just don't understand how, like, after you've denatured it and everything, you're going to look at it and study it if it's all lost its shape.
Okay, so the question is here, how can you study that protein once you've denatured it and pulled it apart and done mercaptoethanol and boiled it, all this good stuff? The answer is you're completely right. You can't. This is just a small fraction of what you have here. And so you'll take a little bit of that, and in that little bit you'll say, oh, look, there's just one protein which is in there. Then you take the rest of it and do all of your fun stuff with that. Yeah?
So just to follow up on that, how does the binding, so I'm assuming the active site is unaffected by these detergents and whatnot so that it can still do affinity binding. Is that okay?
Okay, so again, maybe I didn't do a good enough job of explaining this. Sorry. So basically what you're doing here is it's not everything which is in your cell extract. It's not everything that's in your supernatant. It's not everything that's in this fraction. It's a very small amount of that. And so basically what you want to do is see what is in here, but just take a small amount and put it on the gel. So you take 1%, for instance, and denature it in SDS and put it on the gel. But the other 99% you'll use for, say, your next column chromatography step, or X-ray crystallography, or DNA binding assays, transcription assays, you name it. Yeah?
So you're ripping apart the structure of the protein, right? You're taking a bundle of rope and you're straightening it? Is that what's happening in this process?
That's so the pulling apart the protein and breaking it apart into little pieces, or if you're doing if they're cross-linked with with disulfide bonds. Yes, you're doing that, but you're doing it on a very small amount of your protein. So it's just to see it, to visualize it using SDS page. So with that particular protein, if you pulled it apart, you've blasted, you've heated it in SDS, basically it's boiling. The way you do this, you take and put it boiling water for 5 minutes with detergent and reducing agent. It's highly unlikely that protein's ever going to be active again.
So that's not the same sample that is being used in the affinity.
So it's the way that you know which of these samples here. So we've got a sample here, you got a sample here, got a sample here, got a sample here, got a sample here, etc. You'll take a small amount of each of those samples, heat it up in SDS, put it on the gel, and you will see that one of those samples, or in this case 4 of them, look like this. But again, it's only 1% of those that you've used. You'll take the other 99% and put it on your affinity column. Yeah?
Okay, so ion exchange, is that just separate by charge or charge and size as well?
So the question is, does ion exchange do just charge or charge and size? It's just charge.
And then gel filtration is size.
And then gel filtration is separation based on size, yes.
Affinity is based on what it binds to.
Affinity is based on some specific interaction that it will have, yes, what it will bind to. Yeah?
Is this, what's the main photo there, is that considered an SDS page?
So this is an SDS page gel, yes. Okay, other questions on this? Again, it's all a process to get to finally something where you can play around with what you're interested in. There's a slightly different process where you can separate proteins again based on size. This is something, also not size, charge, not size. Separation based on charge. It's what's called isoelectric focusing. And so this process is what you can do with this really cool toy right here, is separation of your proteins in a pH gradient. Now, pH, that's of course, a negative log of your hydrogen ion concentration. But at any particular pH, the side chains on your amino acids are going to be charged or not charged, and they'll get to a certain position where they have no net charge. And so at a low pH, your protein is positively charged. What will happen if it's in one of these containers or some kind of apparatus, it can now move to a position where it is no longer charged. In this case, it's going to be pH 7. So you have a mixture of different proteins that you have in here. And you also put them in an electric field, positive charge, negative charge. And as always, your negative is going to push away your negative, your positive is going to push away your positive. And then these will migrate to a point where they have no net charge. And if they have no net charge, then they're not going to move anymore in the gradient. And so this is another way to purify things. Turns out it's a real drag, but at least it's a way to purify things. And one of the big problems you have is a precipitation problem. Why a precipitation problem? What's precipitation? That's where proteins like to associate with each other rather than with water. Well, if they have no net charge, then they're not going to be associating as well with water. So they have a tendency to precipitate at their so-called isoelectric point, isoelectric at the pH at which the surface charge is equal, negative, and positive. So people actually don't use this very much anymore. This is, in fact, a piece of equipment that I found abandoned in an old lab. But they do use it for 2D gel electrophoresis. When we talked about 2D gel electrophoresis, again, way back when we talked about different proteins being expressed by different cells, even though they have a very similar genome. This is what's called two-dimensional electrophoresis, which has isoelectric focusing on one axis and SDS page on the other axis. Remember, SDS page is separating just based on size because you've included the detergents and denaturing agents here. So you're separating based on size here. Here you're separating based on net charge of your particular protein. And what you end up with is a whole bunch of spots. And each of those spots represents something with a particular molecular mass and a particular charge. Now, often, and we looked at this before, you can have something with an identical molecular mass but a very different charge. That can be, for instance, different phosphorylation. You've got one phosphate group on your molecule, will be different charge than 2 phosphates versus 3 phosphates versus 4 phosphates. And so those will be different relative to this. How do you do these kinds of experiments? Well, first you separate based on isoelectric focusing. You turn that 90 degrees, and then you do separation based on SDS page. Same thing is true here as you have with your SDS page. You still have to be able to detect those individual proteins. The way this is very often done, and in this particular case, it's using radioactivity. So here, they're radioactive amino acids which get incorporated, and then you put this gel on a piece of X-ray film, and all the spots represent where you've had radioactivity. And you can literally go through, and in fact, this is what people will often do. And with this gel, you can cut out this little spot and identify it with mass spectrometry, which we'll talk about in just a second here. Another way to identify your favorite protein in all of these different spots here is to use an antibody that will react with one particular protein, or for that matter, maybe a tag that you added to your proteins. So this is an example of just a small piece of one of those two-dimensional gel electrophoresis stained here with all the proteins which are there, and now an antibody which reacts with phosphothreonine in this case, phospho-T. And what happens is all the proteins that have a phosphorylated threonine will light up, i.e. they will interact with this antibody here. And so this is a way you can identify which of all these proteins in this whole big mess up here have phosphorylated, and specifically phosphorylation at a threonine. So this is one kind of blot. We'll talk more about blots a little bit later on. Basically, this is a copy of what you had on your gel. Once you've identified a spot that looks like it's interesting, okay, it's interesting because it's got phosphorylation on a threonine, how do you determine what the actual protein is that's there? And that brings us to probably the most exciting, I think, process used in protein determination these days, called mass spectrometry. Mass spectrometry has developed amazingly in the last 15 to 20 years. And basically, it used to be, oh, we got something big and something small. Now you can literally identify down to fractions of atomic units exactly how big any particular piece is. And so what this does, it allows you to take a protein of interest, so usually this would be one of those spots on the 2D gel electrophoresis, break it into pieces with a protease, usually trypsin. And so we know where trypsin cuts. Trypsin will cut your amino acid chain after lysines and arginines, gives you separate peptides, different pieces of your protein. And you can identify the exact mass of each of these peptides, and the mass spectrometers are precise enough that you can identify then this protein based on the mass of the peptides that come off of it, which is a really pretty amazing process. So you have a spot on your gel, you cut out that spot, you treat it with trypsin, put it in your mass spectrometer, and then analyze what the sequences are, and you can go back and identify the protein you had in the first place. Yeah?
Is it keeping them in order?
So the question is, does it keep those peptides in order when they're cutting them up? No, it doesn't. They're just a whole crazy mixture of different peptides. But the precision of the mass spectrometer is such that you can say this one is 1,328.75. And that only gives you a certain set of amino acids that it could be. It's really amazing. Again, but it's the technological progress that's happened with mass spectrometers that allows us to do that. On the other hand, there are what are called tandem mass spectrometers, which will in fact separate those individual pieces from each other, not in order, but it will take, say, one of these. So your first mass spectrometer will give you 4 different peaks. They could be in completely different order than this. And take one of those and break it up into even smaller pieces. And in that process, you can actually generate protein sequence, because in the mass spectrometer, you will break the peptide bond. And breaking the peptide bond will give you the individual amino acids. And since we know the exact mass of individual amino acids, then you can separate them based on each other. There is, however, one problem with that. Leucine and isoleucine have exactly the same mass. So that you can't, in fact, detect in this process. That's why they didn't use that particular example here. But here, if you don't know the sequence of, usually in this case, it would be all the proteins. We know the sequence of the human genome. So we know the sequence of all the proteins that are there. So just in identifying the peptides, we can say which protein this is. If, however, we're looking at the golden mole, anybody see the golden mole? You know, all that good stuff, awards that my lab got? No, you don't care. So the golden mole, there's no genome sequence. So if you want to figure out what peptide this is, you can't compare this to all the known masses to all the known peptides, which are like you had in the human genome. Now you actually have to break this down into these individual amino acids to figure out what that peptide is. Yeah?
So on the left side,
Mm-hmm.
like trypsin cuts after lysine and arginine, you might have one of those bands that's like 6 leucines.
Mm-hmm. Yep.
But on the right side, you can separate each of those leucines into their individual leucines or something?
So it's in fact, so the question is, can you separate them based on where the trypsin is cutting? So it's lysines, not leucines, that it cuts at. Maybe I misspoke there.
Saying that it only cuts after these 2 amino acids. So
Right.
what happens when there's
So if there's a whole stretch of those amino acids right next to each other, you're never going to see those because they've all been chopped up. They're going to be individual single amino acids. A single amino acid, you can't tell what it is related to. So the key here is that you have some that are not single amino acids. And in the best case, you actually will have multiple peptides here, which will correspond to multiple different pieces of your protein. So if you just have one peptide that matches your protein, that'll give you a certain probability that you've identified it. If you have multiple peptides that are matching your protein, that means it's much more likely that that's your particular protein. So this is, people talk about proteomics, looking at all of the proteins that are produced in any given cell at any given time. All of that is done by mass spectrometry these days because it will literally give you positive identification of any of these individual proteins. So that tells you what the protein is, but it doesn't tell you what the protein is doing necessarily. So that usually leads to the next step, which is trying to determine the atomic resolution structure of any given proteins. There are really sort of 2 main ways that people do this now. One is X-ray crystallography. This is sort of the gold standard. Actually, no, there are 3 ways that people do this, but start with these 2 ways, the main ways. X-ray crystallography, you make a crystal of your protein. This is the magic part of this process. You have your purified protein. You've got this big band on a gel, says, hey, it's wonderfully purified protein, and then you send the poor graduate student away for 3 years to try and make a crystal. And sometimes they do, sometimes they don't. If they do, it's fabulous, and you can get these wonderful diffraction patterns. From diffraction patterns, you can generate what's called electron density. It's where all of the X-rays have reflected from each other. And then, given that you know the sequence of this, you can build a model. And so all of these pretty pictures, all these diagrams we see in the textbook of different proteins, they all come from fitting a model to your electron density. And depending on how good your electron density is, you know, how clearly you can delineate where any of the individual amino acids are, it's easier or harder to build your actual model here. Usually, really good electron densities are at about 3 angstrom resolution, 2 to 3 angstrom resolution, and that way you can see all of the side chains of the amino acids. Like a tyrosine, you can literally see where that's going to go. So X-ray crystallography, if you can get crystals, and that's a huge if. Another way to go about doing this is so-called NMR spectroscopy, and this is a very different approach. Instead of getting a crystal, now you have a protein, again, very important, you have a pure protein in this case, in a test tube, and a very, very skinny test tube at that. Has to be at pretty high concentration, which is the main problem with this technique. And then you do NMR spectroscopy, where literally you're looking at where each of the hydrogen atoms are here in this protein relative to all of the other hydrogen atoms that are in this protein. And I don't know how many of you have done NMR spectroscopy, one-dimensional NMR spectroscopy. We can look at splitting of various different peaks, etc. This is basically one-dimensional NMR spectroscopy here and one-dimensional NMR spectroscopy here. What it gives you, if you read one of these, which I have no idea how to do it, but each of these spots is going to represent a distance constraint, so how far one particular atom is away from a different atom. And then you make a humongous table of all of these distances, and then you look at, with your known protein sequence, how all of those things can fit to all of these distance constraints. So you say, okay, there's a 2 angstrom distance between this atom and that atom. There's an 8 angstrom distance between this atom and that atom. And so basically it's a 3-dimensional puzzle that you need to put together based on all these distances between the individual atoms. Yeah?
What does NMR stand for?
Nuclear magnetic resonance. But very few people use NMR. I made that whole name. Yeah?
So this is almost like a sketch of best fit, given that.
Yeah, so this is exactly, this is a best fit to all of the data over here. And one of the things that you'll notice here is there's actually a really pretty good best fit over here on this side and a pretty crummy best fit over here. And that tells you that this part of your protein in solution in that test tube is actually flopping around. And so it also gives you information on what part of your protein is floppy and what part isn't, which is really cool because often that'll be something that's interacting with some other protein. So that's a great technique if you can get high enough concentration of your protein. The other thing is this only works for relatively small proteins because what will happen if you have a big protein, this will be one big black blob. So you can't get that resolution information out of that. So those are sort of 2 ways to do protein structure determination. A much easier way to go about trying to figure out what your protein is to blast it. Oh, I just hate blast, using blast as a verb because it's actually not a verb. So anybody know what BLAST stands for? It's actually an abbreviation. How many of you have used BLAST? How many of you know what it stands for? That makes me. So it's a computer algorithm. It's the basic local alignment search tool. I will not ask you that on an exam. But basically what it is it's a very fast way to search your favorite protein up here at the top, your query, against all known proteins in the database, which, considering that's trillions of proteins at this point, is really pretty amazing that you can do this. And in fact, a lot of the fastest supercomputers in the world are doing exactly this. It's just comparing your favorite sequence at the top to all of the other sequences in the database. So one of the things that you get with a BLAST search like this are a couple of different pieces of information. The first one here is this so-called expect value, e to the minus 111th. This is a statistical measurement about how likely these 2 things would have come up at random based on the size of your database. E to the minus 111 is a really small number. So it's highly unlikely that this is a random match that happens. And partly you can see is because lots of the sequences here are identical. Everything that's in yellow in the middle are identical. The pluses mean that it's a similar kind of amino acid. You have a lysine versus an arginine, so these are similar kinds of sequences relative to each other. So this is a great way, if there is a protein that there's a structure known for, your favorite protein has an E value to the minus 111th, 68% identical at the amino acid level. Anything above 30% is going to have a similar kind of structure, probably a similar kind of function. This is a heck of a lot easier way to go than purifying your protein, trying to get crystals, NMR, etc. So we'll talk about the 2 hybrid screens and the pull downs next time, and all the rest of this stuff as well. But now I will leave you to your course evaluations. Those of you who have computers, iPads, phones, etc., please do that. It's very useful. If you would please go ahead and do that now. If you've already done it and you want to find me in office hours, I'm headed over to Science Building 2 right now.